Well, thank you very much, and I really appreciate this opportunity to talk with the committee. Where's the clicker? Ah, great. Okay. One of the things I wanted to do today, and this is based on conversations I had with Amy Geller and Dr. Vermoon, is give the committee an idea of one of the things they particularly asked us, how do the agencies work together? And then what do we see from NIAID as some of the challenges in product development? And then what are some of the key developments in both vaccines and therapeutics? So is it just this little red button? The green one. Okay, thank you. So that's kind of the outline of what I'm going to go over today. And in this time, I've got quite a few slides, some of which I'll just go over very briefly. They're for your information because I thought it would be helpful. And then I'll focus on what I think are maybe some key points. So one of the things is if you look at NIH, CDC, and FDA, they're all three agencies of the Department of Health and Human Services. And actually our agencies have a lot of discussion on interaction with each other, but we each have different responsibilities. In that NIH is responsible for a lot of research and development, FDA for the regulatory approval, and CDC for a lot of the surveillance and detection. But it's a feedback loop on many of these things, and I think that's an important thing. As I said, NIH, and we are the National Institute of Allergy and Infectious Disease, so we're the infectious disease component of NIH, which is actually 27 centers and institutes. And we have a dual mandate. One is to develop, to keep a pipeline of basic research that informs product development, and the other is to respond to emergency threats, such as Zika, Ebola, some of the influenza outbreaks. Along that lines, a lot of the basic research is designed to feed into product development. And the areas that we're going to focus on today for STIs are the therapeutics, diagnostics, and vaccines. As some of you may have seen, it came out the day of your second meeting here. Dr. Fauci, who is head of our institute, renewed our commitment to this area of research as we go forward. So I thought it would be helpful for people to understand what's the scope of the research problem, our program. And this lists all the organisms that our branch is responsible for in the STI section. And the main point is it's not just single etiology-based infection, but also some multi-etiologic-based syndromes, such as bacterial vaginosis. It's color-coded, in red are the 5 that receive the most amount of funding currently. So the goals of the program are multifaceted. I think this is important. It speaks to some of the things that Ilya Vabar and Emily Aioli and others have talked about, is what is our long-term outcomes. And what we want to look at is preventing burden of disease, preventing cancer, reducing HIV transmission, supporting young people, particularly adolescents. And I think a new thing that people haven't thought about is the combating antimicrobial resistance. Everything that we do now in NIAID is mapped along the product development pipeline. This has been, I think, an evolution of the institutes over the last 10 to 15 years that was spurred by the biodefense initiatives after the anthrax outbreaks around the country. And so all of our programs are based along this pipeline, leading to how can we go through the basic research. And what many product developers would tell you is the 2 valleys of death, which is getting a product out of a laboratory into a clinical study. And that's one of the focuses for our gap-filling activities. And the second valley of death is that first-in-human clinical trial, because that's your largest risk from a product development point of view. And so those are the two of the areas that we partner with companies who are developing products of public health interest to try and de-risk those valleys of death to move things forward. And I think you've heard one very good example that I'll talk about in a little bit more is Zoloflozin. So how we set some of these research priorities is to maintain this pipeline, but then also to fill these critical gaps. And we, doing that, do have to focus on what is your critical pathway for licensure. Because ultimately, none of your public health agencies are manufacturers. And our goal is to provide the basics for what you need to make these products with the hope of then a commercial manufacturer or a nonprofit such as GARD-P will take that up for manufacturing and distribution. We maintain strong collaborations with our other partners, and I just have a lot of the logos here. It's not just the HHS agencies, it's also our colleagues at the Department of Defense, as Walter Reed Army Institute and Uniformed Services, as well as internationally with the World Health Organization, who we work with quite extensively, as well as some of the foundations, the Wellcome Trust and the Bill and Melinda Gates Foundation. So that just gives you an overview of our philosophy and our programs. I'll be happy to answer any additional questions on that. But today I wanted to focus on three particular areas because I think they're of particular interest to the STI community. And I will tell you the antimicrobial resistance has certainly changed our dynamic about how we think about particularly Neisseria gonorrhea. Many of you are probably familiar with the President's National Action Plan for Combating Antimicrobial Resistant Bacteria, which is called the CARB plan. The component that, of course, NIH is very involved with is the research component. But it has also changed our dynamic in what we view as some of the priorities for products. And that would be reflected in a vaccine, say, for example, for preventing Neisseria gonorrhea. So let me focus on that organism for just a few minutes. As you've heard, from other speakers and from many of with your research colleagues, Neisseria gonorrhea was identified as one of the top 3 urgent threats by our colleagues at the Centers for Disease Control. Part of the reason for this is the ease of this organism to develop antibiotic resistance. And this is a slide that I think every gonococcal investigator uses to show what is the time frame. And the important thing here is when you look at this, you can see that the time frame is collapsing for how long an antibiotic is actually of clinical use. And unfortunately, we're to the point now, actually, that we are down to 1 antibiotic being recommended by the CDC, which is ceftriaxone, for common syndromic use and management. One of the things we do is support basic research in trying to understand what are those mechanisms of resistance and what are the types of things that could be new drugs. To that extent, one of the things you've heard from the GARD-P from Emily was that Zoloflozin was a first-in-class drug. It's a non-quinolone topoisomerase that is orally delivered. And as Mano said, we started working with Infectious right at the beginning. When they approached us, this was not high up on their company's development plan, but it was one that was only being developed with gonococcal indication. Just as an aside, to put it in perspective, most new antibiotics for Neisseria gonorrhea, that is not their primary indication that the companies are developing in. In fact, it's usually for anthrax, tularemia, things, because they're coming out of our biodefense program, which is the other antibiotic that is currently entering a phase 3 trial, which is geptidiosin from GlaxoSmithKline, which was supported by BARDA, but for biodefense. So we entered into a partnership with Infectious through our clinical trials grant, that's contracts, that's at the far end of that product development pipeline, and conducted the phase 2 trial to look at early proof of efficacy to de-risk the product. And this was actually even before the company had their final formulation. It was administered as an oral suspension, which is really unusual. In fact, most people said, Carolyn, you're nuts to think that you can do a product development in that order because it's not classical. And so when people were asking some of those questions, that's some of the risk you take to accelerate product development. Luckily, it was very effective. And so that's what you are hearing from Emily is now what all has gone into getting to that phase 3 trial. Because then in addition, from the phase 2 that showed efficacy, you had to have your final formulation and had to do a PK/PD bridging study to show that the final formulation was the same that was in your phase 2 trial. And then this is handed off by NIAID to GARD-P for the evaluation on a worldwide basis that I won't go into because Emily covered it quite well. So that's one of the ways we look at developing new antibiotics, in particular for addressing AMR. The other is in diagnostics. And can you use diagnostics in addition to identifying the source of the infection? Also say, is that infection susceptible to treatment with an older antibiotic? For Neisseria gonorrhea, this works well for ciprofloxacin because it's a defined mutation. And so you can develop a nucleic acid amplification test to do that. But one of the things the FDA potentially will ask you is to demonstrate its clinical use, not just feasibility in the laboratory. And so currently, Jeff Klausner at UCLA is doing a clinical study to show that if you say the patient can be treated, it will be an effective treatment. And we're hoping that that will help then the development, and there are now several other companies that are trying to incorporate this into their devices. I think one of the last things in this area, and you heard Laura this morning talk about the extragenital testing. This was actually a challenge to get this FDA approved and had been identified probably 10 years ago as a public health need. But from the manufacturer's point of view, that was not something that was worth their investments. So NIAID, in collaboration with our antimicrobial resistant leadership group, partnered with 2 of the diagnostic companies to run the clinical study that obtained the data to be able to allow those companies to submit their 510Ks for approval of those devices. And I think that was something that was a unique public-private partnership came from CDC. We figured out how to respond. We worked, it took 2 years, by the way, to get the clinical protocol agreed upon with our colleagues at the FDA. And they deserve a lot of credit for being flexible and thinking outside the box. And Charlotte will give you more info on this if you are willing to talk to her. But I wanted to make the point, this is not NIAID only. It goes across institutes. And one of the new diagnostics was originally given seed money by a NIAID and NIBIB grant to Charlotte Gatos. Let me turn now to vaccines. That was a whirlwind through AMR. This, in my mind, is how we are going to potentially prevent the eventual, spread of these infections and have eventual control, is vaccine development. We've been working with the WHO, STI, and Reproductive Health Group now for over 5 years to develop a roadmap. This is also in partnership with the WHO Product Development for Vaccine Advisory Committee. But one of the things Sten asked me to talk about, and Ilya Barr did, is why is this so difficult? This is all the stair steps in how you develop a vaccine, from basic research all the way through your development, your clinical evaluation. And it looks like a one-way street, but it's not always. And many times you fall back and have to go back up the steps over and over again. You notice this has a 10 to 15-year time frame. It also has probably anywhere over $1 billion to in excess of that $1 billion cost to development. And you notice there's no guardrails on these steps. And so for many times, people just fall off the steps, and many promising products, unfortunately, never get to a phase 3 trial. The average success rate in a phase 3 clinical trial is 1 in 5. So it's a huge financial risk. What we try to do is keep the pipeline going. For herpes simplex virus, there's a variety of approaches, from subunit vaccines to live attenuated to DNA vectored vaccines. And again, not only I'm in the extramural side of NIAID, but the intramural side has recently completed. Jeff Cohen, our colleague in the intramural side of NIAID, has finished a phase 1 study of a vaccine for therapeutics for HSV infection in collaboration with Sanofi Pasteur. The gonococcal vaccines, there's again a pipeline of development, similar types of things, subunits, outer membrane vesicles. You notice we try to keep our array, because think of like a funnel, and you want to start out with lots of things in that funnel and evaluate and narrow down till you get to your final product. We've had very encouraging things here, that there may be a proof of concept for a gonococcal vaccine based on outer membrane vesicles that came from Neisseria meningitidis, which is a closely related organism. This is animal data from Ann Jerse's lab at Uniform Services. And what you can see, mice immunized with Becks Arrow generate antibodies that react with gonococcal proteins on a Western blot. And they also reduce colonization in her animal model. So one of the things, in addition, this vaccine was introduced to control an epidemic outbreak of group B meningococci in New Zealand. And a nested case control study retrospectively showed that you had a decrease in Neisseria gonorrhea infections of 33% compared to none for chlamydia. This vaccine is licensed in both the U.S. And in the European Union. It's Becks Arrow. It was developed by Novartis, which was bought by GlaxoSmithKline. And we are currently exploring doing a prospective clinical trial to demonstrate efficacy of this vaccine for preventing Neisseria gonorrhea. We have refocused a lot of our efforts from our STI cooperative research centers on vaccines. These are the 6 recently awarded STI cooperative research centers, 2 of which focus on Neisseria gonorrhea, 2 on chlamydia trachomatis, and 2 on treponema pallidum or syphilis. And we're hoping this will provide us an opportunity to focus on product development with these groups over the next 5 years. So the last thing I want to briefly touch on is therapeutic options. You heard from the developers there needs to be a pipeline, but this is actually something that is risky. Through our, we have a variety of preclinical contracts, and we'll entertain if a company has an antibiotic that they're developing for another indication, we will do the screening for them against a panel of approximately 100 Neisseria gonorrhea strains that was obtained by the CDC surveillance program and is well characterized for all their antibiotic resistances. What that does is potentially allow them to consider a gonococcal indication as they are developing their new products, and as a way to de-risk that product development. In addition, I did want to touch on those other non-single etiologic organism syndromes, particularly bacterial vaginosis, which is a large problem for many women worldwide. I think this is something that has been a challenge because of the endpoints in clinical trials, but it's also, I think, an opportunity where we have learned from the human microbiome and our understanding now of the vaginal microbiome that there may be a way to repopulate the vagina after treatment with a live biotherapeutic. And I wanted to talk about this because this is a new class of products that many people aren't that familiar with, but it is great interest in the community, both this and for C. difficile for treatment of recurrent C. diff infections. And this, we have had ongoing discussions. There is not a clear regulatory pathway for these products. And so this is something we are in active discussion with our colleagues at the Center for Biologics at the FDA, where they go for regulatory consideration. We do, and the other point, just to make the point, things that come out of the CDC treatment guidelines meeting all the time raise new research questions. And so that's one of the things. NIAID participates in those meetings as a liaison and then can embark on clinical studies to try and elucidate some of those questions. And again, back to our original roots is keeping the basic research going. For those of you who have worked or heard of people who work with Treponema pallidum, you know that maintaining culture was impossible. You culture this organism in rabbit testicles. A recent breakthrough has been the potential to culture treponemes in tissue culture. And we're hoping this opens up a whole variety of new product development opportunities. So with that, I'd like to thank you, the people in our group at NIAID, all my collaborators at various parts of NIAID. We are only one of the institutes that funds STI research. It's also funded at NCI, for particularly HPV, the National Institute of Child Health and Human Development, and the National Institute of Mental Health, as well as others. So thank you all very much. And I tried to stay to my 20 minutes, Tim. Thank you very much.
Excellent. Grab a seat, Carolyn. Thank you very much. And we'll take any questions from the audience. Mike.
Thanks, John. That's great. Just a quick question. DMID is in the throes of reinvigorating their research portfolio right now. I don't completely understand why the gonorrhea project went to GARD-P and what the future holds for research in general from DMID. What was the basis for, because you could have done it yourself. So.
Right. So I think the gentleman from Infectious talked about that. One of the things we try to do is get it to a point where the manufacturer will move forward, or if it's a biotech company, that they will be able to successfully compete and obtain venture capital. Okay, so you give them, we hope to provide enough data to do that. I think for many of the larger manufacturers, initial data will allow them to develop it, similar as you heard Ilya Barr talk about for Gardasil. That original study he talked about from Lorikowski that showed that original epidemiology of HPD was a NIAID-funded study. So I think it depends on which companies you're talking about, and the landscape may be very different from what you could traditionally think as large pharma versus a biotech. And so the idea to go to GARD-P, which was able to secure, Emily talked about where their funding came from. You notice it's all from a lot of countries that are interested in this area, from some of the foundations that are, and it will allow the further development. That trial is going to be a fairly involved clinical trial. It's in 4 different countries for licensure in 3 different regulatory authorities right off the bat, the FDA, the EU, and the South African Medicines Control Board. And so I think having that coordination by an entity like GARD-P, which is a spinoff of the Drugs for Neglected Diseases Initiative, provides that regulatory support also.
So I was curious if you have any analysis of the level of funding NIAID has for STI vaccine or therapeutic development, and how it compares to like the disease burden for STIs versus other diseases? Like, what are your thoughts on, are you adequately funded, underfunded? How do you judge that?
So that's a very difficult question, because I think one of the things as you look at is that changes on a fairly rapid basis depending on how the diseases are occurring. I did anticipate that question, so I asked our budget office to run the numbers for it. In 2018, in NIAID, it was approximately $200 million of funding for what's coded as STI research. But then there is significant funding because HPV is probably more funded in the National Cancer Institute. And so I think overall, if you look at NIH as a whole, it's probably about double that.
Earlier in the previous session, it was commented that you've got the EMA for Europe, the European market. You've got the FDA for the U.S. market. You have the MCC for the South African market. You've got the China FDA for the Chinese market, and on and on. Is it seen as a priority by the U.S. Public Health Service to help the FDA synergize across these various markets? The cry of help from the private sector is clear, that segmented markets discourage carrying products that are not maximally, profitable through to developing country use or to broad market applicability. They're more likely to license in Europe, make their money, and be done with it, or in the U.S., or simply to abandon a product if it just doesn't make enough money.
Yeah. So I think the short answer is yes, particularly in the area of vaccines. If you talk to our colleagues at the Center for Biologics at the FDA, they've been involved for quite a few years with the ICH effort, the International Committee on Harmonization. That's been a way that I think the regulatory agencies discuss with each other how they could harmonize some of those requirements. I think the other group that we discuss that with is our colleagues at World Health. When you look at what is the, and that was something that SAGE, of course, is the recommenders at World Health for what vaccines go in the immunization program. But they started an initiative several years ago, I think about 3, the Product Development for Vaccines Advisory Committee, that does horizon scanning on what is coming down the road once things are in clinical trials. So I think all the public health agencies worldwide realize that's an issue. You heard one of the comments from Ilya Bar about the manufacturing plants. And I think that is one of the emerging areas that people are realizing. A manufacturing plant could have 5 different inspection teams. You could have an FDA one, a WHO pre-qual, an EMA. Can you start in this? I know there's discussion started. Can you have reciprocal information sharing about those inspections? And that extends to the low and middle income country manufacturers. I think just as a comment that maybe is a bit of an aside, when you heard the cost of a manufacturing plant, a lot of people don't realize that a biologics license is not just for the end product. It's also for the manufacturing facility. And the same thing, WHO, when they pre-qualify a product, look at the facility. As you move things into, say, India, or those manufacturing plants have to maintain the same quality assurance as any US or EU-based manufacturer. And we're even hearing now through the decade of vaccines discussion that you're reaching a floor that people cannot go below for pricing because of the need to maintain those buildings. And I think that's one of the considerations that's going to be discussed as we go into the next decade of vaccines between NIAID, WHO, the Gates Foundation, about how we address those implementation manufacturing issues. Can I answer
yes,
the one
please,
Can I answer the one that you sent me by email? Because since I spent most of last week agonizing about it.
Thank you.
So the question that he asked me, and it's actually a point I wanted to make, is if I had 3 key wishes for STI, see, you'd forgotten, you'd asked me that, what would they be? And so I think the first one is vaccines. I really think that treatment will not, in the long term, control this problem. It has not historically for any, and we have to think about that. I think the dynamics now have shifted, and certainly many of these vaccines could be part of an adolescent platform, building on the HPV vaccine, the meningococci vaccines given in adolescents, and others that are coming down the pipeline, such as for Group B strep. I think number 2 is better epidemiology, because I think right now we don't fully globally understand the problem. And that creates multi-layered problems, because if you don't understand the depth of the problem, the extent of the problem, you cannot do the economic modeling necessary to say why these products could be effective, cost-effective, and therefore of value for investment. So that would be my second. And we're trying to do that. Carol Jimenez is here. She's leading the federal STI action plan you're going to hear. That's one of the things to see how we can address it. The WHO has been addressing areas like how you can improve surveillance. And the CDC certainly has led this way. And my final thing is I think we need to make these infections like any other infection. In other words, people get an infection, they either get an asymptomatic or a symptomatic disease, and you want to treat it or prevent it. And as we move forward in this area, I think if we're going to improve adolescent health and look at these areas, that is something that has to be incorporated. And all of this, and your all's committee's work could certainly help add to that as a coordinated effort along with the STI Federal Action Plan from the U.S. government. So thank you very much for letting me say that.
We do have a final question from Mike, and then it's lunch.
Just really quickly, one thing you and I have spoken about before is your concern about the stigma associated with STI vaccines, that the HPV vaccine is a cancer vaccine. My question has to do with the target population for STI vaccines, because these STIs kind of evade Mother Nature. So theoretically, I guess, if you made, it's a question on a statement, if you made these vaccines, the target population could be people who had an STI who are at graver risk for reinfection. Is that correct in your thinking?
I would probably disagree with you on that one, Mike.
That's why I asked.
Yeah, so, because we've had this discussion. So I think one of the other things, and I've brought this up in other settings, other people, many of you who know me have said this, I think we do the field a disservice by saying STIs. Actually, they are different infections. So I would say you could potentially track gonorrhea and chlamydia together. Okay, there's your typical broad use. You even heard Ilya Barr say chlamydia. Okay, that all fits in an adolescent platform potentially. Herpes is the same thing. It could be something very different. Maybe it's different for syphilis, maybe not. I don't know. But I think we do a disservice for lumping them together rather than talking about them as specific infections. And the other reason, from a product development point of view, how I develop a product for a viral vaccine is very different than a bacterial. How I make an antiviral is very different than how I make an antibiotic. And maybe your point of care test would be very different. And so from a product developer's point of view, in a research point of view, I consider them individually, not as STIs. And I hope the field somehow figures out how to convey that. This is not one infection, it's a variety of infections, each of which is unique and different.
Interesting perspective. Thank you very, very much.